Today is the 1st of July 2025, and today we are going to learn about microbial resistance to antibiotics. Many isolates of microbes are resistant to antibiotics because they acquire what is known as plasmids. And these pose a significant threat to public health and safety, because if a microbe acquires resistance to antibiotics, the antibiotics which are administered to you by the doctor will not function against that particular microbe. So this is called antimicrobial resistance. Now, to determine antimicrobial resistance, we do a specific test using a specific microbial medium. This microbial medium is known as Mueller Hinton Agar. And as you can see on this particular Petri plate, we have poured it to a depth of exactly 5 millimeters. This is the standard depth for Mueller Hinton Agar. Then we spread the isolate on the plate using a spatula, and we place what is known as an antibiotic disc. This is an antibiotic disc, which is a standard. Now, these antibiotic discs, I will show you an example over here, are impregnated with specific kinds of antibiotics as indicated on the code. Now, this is a lawn of growth. This is sample number 7FG. It's from sample collection point number 7, and FG is the code which I have assigned to it. As you can see, this is Gen. If I look back at the code, Gen is gentamicin. Now, this microbe is susceptible only to gentamicin because there's a zone of inhibition around it, whereas the rest of the antibiotics, there is no zone. There's something over here, but that's very minor. Let's look at this same strain number 7FG with another set of antibiotics. If you look closely, no inhibition. The microbe appears to be growing freely in the presence of these antibiotics. So that tells us some very interesting information, or it informs us about this particular microbe, and we'll go into that later when we analyze the data. Now we look at number 5W. Let's look at 5W closely, and we can see, yes, it's resistant to all these antibiotics. We'll note that down in our results later. And this one, again, gentamicin, rest resistant. Okay, let's look at the next strain here. So we have number 5Y. Now I've given the code Y because it's yellow. Let's look at it closely. Okay. Again, we see it appears to be growing quite well with the exception of this particular antibiotic. I will check the codes later and we'll record this result. So there is a zone of inhibition around this particular disc, but it's not significant because it's quite a small zone. Then we look at the next one, which is 5Y, but with a different antibiotic. And then you can see here, look, and this is VA, which is vancomycin. So vancomycin can inhibit this particular microbe. The rest, let me look closely. So you see gentamicin, and we have E. So I'll check these codes later, and we can see that these antibiotics are working and they are reducing or inhibiting the growth of this particular bacterium. Let's look at 4R. Now, the code number 4 is the sample site. R is because this one was red. What's surprising is that at 37 degrees Celsius on Mueller Hinton Agar, it does not show the red characteristics. Now, as a microbiologist, this will tell you additional information about the strain, which I will discuss in another video. Now again, you can see gentamicin, and the rest, almost no inhibition. So gentamicin inhibits the growth. You can see it on the disc here. Okay, now let's look at the last one, which is 6WH. So this is collection point number 6, and it was white. Okay, now this is very interesting in terms of the growth profile. As you can see here, you have some kind of inhibition, here something, here. Now what's interesting is you look at this one closely. you will see that this particular antibiotic has inhibited, but then can you see there's a growth after that? So that means this implies that this particular bacterium has developed resistance because it has started to grow after inhibition. Can you see that? So we'll discuss this later because it tells us some very interesting facts about that particular microbe. Again, 6WH on the next plate and the next set of antibiotics. Again, you can see some kind of inhibition but not much. For example, gentamicin definitely is inhibiting the growth. It's a very strong antibiotic. The remaining, not much. So based on the resistance profiles, we can learn a lot about these particular bacteria and the pathways which they have in their genomes and the mechanisms whereby they resist antibiotics. Now I extended this to another experiment on Mueller Hinton Agar, in which I tried to check how these microbes will actually work against each other. So microbes, in addition to being inhibited by antibiotics, they will also be resistant to exudates produced by certain microbes. Let's look at this. Okay, look at this plate and you can see a clear zone here. Okay, this is the clear zone. You can see it over here. Okay, there's no growth. Now I flip over this plate and I look at that code. So this is number SA7FG. This is the number 7, which is actually this. This bacterium, for some reason, is producing some kind of antimicrobial compound, and that is inhibiting the other bacteria. Can you see it here? So we'll have to do a 1-to-1 assay. Now this contained around 1, 2, 3, 4, 5, 6 strains. But when we do a 1-to-1 assay, we take one microbe versus one plate. This is just a preliminary study. We'll shift it to 1-to-1 plate assay. Now all of these results show something very interesting regarding the antibiotic profile. And as microbiologists, we look at the antibiotic resistance profile both in terms of the clinical implications of that particular assay as well as the implications for the genetics and the production of antimicrobials. Certain microbes can be harnessed for the production of antimicrobials against other microbes. So this is a medical opportunity. So thank you for watching, and I hope you learned something about antimicrobial resistance.